Hello everyone, myself Sneha suman purbia from group 316. This is my topic synaptic transmission. Synaptic transmission it is a process which nerve cells communicate among themselves or with muscles and glands. When the impulse reaches to the nerve fibers endings, it either performs its function at a neuromuscular or a myoneural junction, inducing muscular contraction, or is transmitted to another nerve fibers. In either cases, there is accomplished a synaptic transmission of a nerve impulses. The synapse is a functional intermembrane junction between the two excitable cells. The distance between the presynaptic and the postsynaptic membrane uh, is known as a synaptic cleft. It is varies in different. It varies from 15 to 20 nanometers. In a myoneural junction, the gap is even larger, up to a 50 to 100 nanometers. There are two types or two modes of synaptic transmission, or we can say that synaptic conduction. First is the electrical, and second is the chemical. In an electrical synapse, we can say that the electrical synapse, their synaptic cleft difference is a point 2 nanometer, and in the chemical synapse we can say that their synaptic cleft distance is a 20 to 50 nanometer. In an electrical synapse, neurotransmitter is absent, but in the chemical synapse the neurotransmitter is present. um, in an electrical synapse, the conduction of an impulse is a bidirectional. Either it goes in this direction or it go in this direction. Here the charges are present, but in the chemical synapse we know that there is the release of a neurotransmitter is present. But in electrical synapse, the neurotransmitter is absent. You can also see the diagram of the conduction of the impulses. This is a diagram of the chemical process, and this is the diagram of the electrical process. Differences in the electrical and the chemical synapse. In an electrical synapse, uh, the conduction speed is faster than in the chemical synapse. The chemical synapse is a unidirectional, while the electrical synapse is a bidirectional. Synaptic delay and fatigue is absent in a electrical synapse, while in the chemical synapse the synaptic delay is present, and their conduction speed is fast. Example of the electrical synapse is presence in cardiac muscles, while the example of the chemical synapse is mostly present in the body between the neurons. And the gap junction is present in between the electrical synapse, while the chemical synapse does not contain any kind of gap junctions. This is the diagram of the chemical synapse. You know the process which is occurring in the chemical synapse. um, First the impulses are come, and the depolarization will occur. After the depolarization, the calcium gated voltage channels will open and the calcium influx will occur. This calcium will go and bind to the synaptic vesicles. These synaptic vesicles will move, and the exocytosis of the neurotransmitter in the synaptic cleft will occur. After that, the neurotransmitter will come out and bind with the sodium gated ligand channel which is present on the postsynaptic membrane. Then after this, the generation of the threshold value will occur on the postsynaptic membrane, and the uh, uh, this will induce the excitatory postsynaptic uh, potential, so that the depolarization will occur inside the postsynaptic membrane. After the depolarization in the postsynaptic membrane, Then the breakdown of this extra ACH will occur by the enzyme called SCHE, which will induce the which will convert ACH into acetate and choline. This choline will further go to the presynaptic membrane via the transporter protein. Let's zoom this process into here. The when this choline will come inside this presynaptic membrane. At the same time, the formation of ACH will also occur in the mitochondria. So the acetyl coenzyme A is come from the mitochondria, and this acetyl coenzyme A plus choline, both in the presence of Kate enzyme, that is also known as a choline acetyl transferase enzyme, will convert this into a coenzyme A plus actin. This coenzyme A is also go into a mitochondria and help in formation of a coenzyme acetyl coenzyme A, and this actin is go and help in the formation of refilling, or we can say that in a synaptic vesicles. Now here is a point, that is the in neuromuscular junction the only ACH is a neurotransmitters we use. Let's see the this is the about the excitatory post synaptic potential. Let's talk about the inhibitory post synaptic potential. In an inhibitory post synaptic potential, the process will is same, but in the presence of ACH we have here is GABA and serotonin as a inhibitory neurotransmitter, and in the place of a sodium gated ligand channel we have here the chlorine ligand gated channels. uh, when the neurotransmitter is come out, the chlorine will go inside, and this negative charge inside the cell will the postsynaptic membrane is increases, but here is no hyperpolarization will occur. So let's talk about the types of a neurotransmitters. All inhibitory neurotransmitter induced sleeps, no, this is not occur. Types of a neurotransmitter: first we have a excitatory, and second we have a inhibitory. In an excitatory, we know that the depolarization will occur, excitatory post synaptic potential will occur, and the binding with sodium gated sodium ligand gated channels will occur. Here example of the excitatory neurotransmitter is first SEH, second is epinephrine, third is a norepinephrine, fourth is a glutamate, and fifth is a aspartic acid. In an uh, inhibitory neurotransmitter, we have a GABA, serotonin, dopamine, and glycine. This is also an amino acid. So here is my second topic, the kinetic properties of the muscle. It is a quantitative theory expounded above refers to the steady muscle contraction. Of no less interest are the dynamical properties of the muscle under a non-steady state conditions. The steady contraction has character of a plastic flow. In non-steady conditions, the muscle exhibit elastic properties. For instance, in quick release experiment in which the isometric contracted muscle is released and experience a rapid isotonic contraction, they are observed slowly dumped oscillations. The frequency of these oscillations in is of order of a curves for muscle several centimeters long. If we regard the muscle as a viscoelastic body, we are construct a model containing a damped elastic elements connected in series with an undamped elastic elements, and one more elastic elements is parallel with the first two elements. Such a formal chemical processes model is combined of a Vogit and Maxwell bodies. The the Vogit model is elastic element connected with the parallel with a dumped element, and Maxwell model contains the same element connected in series. This is the diagram of the kinetic properties of the muscle. Uh, the generation of a oscillation in the muscle can be determined by a known linearity of a non-steady state kinetic equation that cannot contain elasticities in an explicit form. The possibility of the oscillation is this case due to the kinetics of the closing and the opening of a cross bridge. On the other hand, the bridge itself is a viscoelastic system. The tension generated by a closed cross bridge may vary in a stepwise manner depending on the angle at which head of HMM and SI arrange itself relative to action, and also the extent of stretching of S2. As you see in this diagram, the the transition between the steps have an effect on a rapid non-steady response of the muscle. Thus the oscillation during quick release are caused by a elastic deformation of the bridge. This is the most probable through yet proved suspension. Of great interest of the physics and biology are the flight muscles of insects, that is known as a IFM, and related uh, tamponal muscles of cicads. These muscles are capable of a rapid period contraction with the frequency of an order of 100 Hz. The flight muscles of insects are structurally similar to the crossing striated muscles of the vertebrates. It has been established that the sliding filament model with the actin and myosin bridge is applicable to the IFM. The rapid oscillation of IFM requires the presence of directly functioning elastic element. Microscopy shows that in contrast to the muscle of vertebrates, in IFM there is a direct viscoelasticity link between the myelin filaments and the Z membrane, accomplished by a special element that we show in the picture. Probably this element is capable of undergoing elastic deformations. There has been established large difference between the oscillations of the IFM and those of the potential transmitted to these muscles. For example, these flies are frequently to the potential transmitted to the IFM is the 3 Hz. The frequency of the wings oscillation reaches to the 120 Hz. Hence, the muscle is not activated with each oscillation. The oscillation of the IFM of the nature of auto oscillations. This auto oscillation arises in a non-linear state due to the force dependent on the state of movement of the system itself, or the amplitude of the auto oscillation is independent of initial conditions. uh, the auto oscillation of IFM are excited in the presence of a feedback between the deformation and the stress. The relationship between the IFM contains the transformer elements with respond to the mechanical events that control the state of the contractile system. This element is localized by myofibril, which is proved the presence of auto oscillation in the IFM. Preparation caused by the glycerol during the twitch of the muscles of the vertebrate there is observed. A characteristic period of the process which is occur at the same sort of the latest fashion is a finest step. This was detached by an optional method that is known as a Frank that is proposed by the Frank in 1964. A very important method of studying the muscular dynamics of the muscle is the rapid X-ray structural analysis based on application of a synchron radiation. Winza and the co-workers are succeed in carrying on X-ray filming the muscle with the resolution up to 0.003 seconds. The kinetic properties of muscles have been studied little. The general approach of the understanding of these properties must be based on the theory of a non-linear dynamical system. This is all about the two topics. First is the neurotransmission and second is the kinetic properties of the muscles. Thank you.